Pasteurella multocida is a gram-negative bacterium that causes disease in a variety of hosts, including hemorrhagic septicemia in cattle, fowl cholera in poultry, pneumonia in swine, snuffles in rabbits, and wound infections in humans. There are many different typing methods that are used to differentiate isolates of P. multocida. Two common methods include serogroup and subspecies identification. Traditionally, these methods have relied on labor-intensive protocols or phenotypic assays for differentiation. Currently, there are no bioinformatic tools publicly available to differentiate serogroups and subspecies from whole genome sequences. Therefore, the aim of this study is to develop in silico molecular typing methods that can be used to differentiate P. multocida whole genome sequences. There are 5 serogroups of Pasteurella multocida, and these serogroups are based on the antigenicity of the outer capsule of the bacterium. It is important to identify the serogroup because specific serogroups display increased virulence in certain species and may also aid in outbreak investigations. The genes encoding these different capsules have recently been identified. The nucleotide sequences of these genes were downloaded from GenBank and used to construct a custom BLAST database. In order to test whether a BLAST search could correctly identify serogroups, all 55 whole genome sequences of P. multocida that had serogroup data available on NCBI were downloaded. All genomes were searched using the customized BLAST database, and top hits for each genome with a bit score of less than 1,000 were removed. Of the remaining top hits, the hit with the highest percent identity was selected. This methodology selected the correct serogroup designation for 54 of 55, or 98% of genomes. This methodology was then applied to a larger set of whole genome sequences. This included 653 whole genome sequences from various hosts and geographic regions. The resulting serogroups were used to annotate a SNP-based phylogenetic tree of the whole genome sequences. The outer rings represent the different serogroups A, B, D, and F. The colors on the inside of the tree represent the corresponding hosts from which P. multocida was originally isolated. Serogroup A was predominant, followed by serogroups F, B, and D. In avian hosts, all serogroups were identified. Bovine hosts accounted for almost all of the serogroup B genomes, but serogroups A and F were also identified from bovine hosts. In some cases, it appeared that the serogroup distinction corresponded to SNP differences at the whole genome level, such as with the serogroup F genomes at the very top of the tree and serogroup B at the bottom of the tree. Ultimately, the identification of these serogroups in silico allows us to assess the relationship between serogroup and other epidemiological factors such as host, geography, and gene content. There are 3 different subspecies of Pasteurella multocida, and these subspecies are based on the ability to ferment sorbitol and galactitol. There were 68 genomes on NCBI with subspecies information available. These genomes were downloaded, and a pangenome was constructed, which identifies all genes present in all of the genomes. The pangenome was searched for genes related to sorbitol utilization. 4 genes that encode sorbitol-specific enzymes were identified, including SRL, A, B, D, and E. However, these genes were present in genomes of all 3 subspecies, including subspecies septica, which is distinguished by the inability to ferment sorbitol. To investigate this further, a SNP analysis was conducted which aligned SNPs found in each of the genomes to a reference sequence composed of the SRL genes. Interestingly, 12 of the 13 subspecies septica genomes contained a specific SNP at position 520 in the SRLB gene that resulted in a premature stop codon. The one subspecies septica genome that did not contain this SNP had a missense mutation at position 226 in the same gene. Therefore, it is likely that the SRL genes are responsible for the inability to ferment sorbitol in subspecies septica isolates. Additional investigation is needed on a larger set of genomes with confirmed subspecies. However, this serves as an initial step towards the development of an in silico method for distinguishing P. multocida subspecies. Establishing computational methods for existing typing schemes of P. multocida will enhance our understanding of the molecular epidemiology of Pasteurella multocida and provide researchers with additional tools for classifying isolates outside of a traditional laboratory setting.